Good day, everyone, and welcome to another screencast here in microbiology, in bacteriology. I am your professor for today, Dr. Supachai Basit. And for today's session, we will be talking about microbial taxonomy. So, it's very important for us to determine or identify what are the different domains of microorganisms. So as you can see here in the illustration, we have several microorganisms. We have here bacteria, we have here fungi, we have here a microalgae, we have here an example of a parasite or protozoa, and here is actually an illustration of T helper cells being attacked by HIV. Okay, so let's talk about first bacteria. Bacteria is an example of microorganisms and they are said to be prokaryotes. So pro refers to primitive and karyotes refers to nucleus. So when we say prokaryotes, this actually means primitive nucleus. So they do not have any organelles and they are considered to be as unicellular. So you can actually see here the difference between a gram-positive bacteria and a gram-negative bacteria. So later on, we will be discussing what are the basic differences between these two classifications. And then we also have parasites. Parasites are actually eukaryotic organisms. So like the one that we studied in parasitology, organelles are present. So parasites means that they cannot live without hosts, particularly those that are considered to be as an obligatory parasite. So they could either be single or unicellular or multicellular. So unicellular include the protozoan in which they can obtain nutrients by means of ingestion. Now, the one that you can see here is an example of a tapeworm that is a multicellular parasite, not bacteria. Okay, so these parasites could either be motile or non-motile. So protozoans, for example, are categorized according to their locomotory organelles. They could have flagella, pseudopodia, cilia, and some varieties. So some varieties have no obvious means of locomotion, such as the plasmodium. Okay, now let's talk about fungi. Fungi are actually heterotrophic eukaryotes, so which means that they can obtain nutrients through absorption. An example is yeast, which is a unicellular. They are considered as unicellular. The one that you can see here is an example of a yeast, Candida albicans, okay, which can cause moniliasis or thrush. Okay, so however, the yeast here is the one that is found in the urine. So how would you know it's a yeast? Because of the budding arrangement. Okay. Most fungi, however, are multicellular. So they reproduce either sexually or sexually. Now, some fungi may be considered as dimorphic, meaning to say their morphology changes depending on temperature. So let's say, for example, Histoplasma capsulatum is an example of dimorphic fungi because at 37 degrees or at body temperature, they are at yeast form, but at room temperature, they are filamentous or mold form. Okay, so eukaryotic cells are like this. So they have true nucleus. So the term eu refers to true and karyo refers to nucleus. So eukaryotic refers to true nucleus. So they have complete locomotory organelles, and the reason why we call it true nucleus is because of the presence of a true nuclear membrane. Alright, viruses. Viruses are the smallest infectious particle, not really smallest because prions are much smaller than viruses, but at any rate, viruses are considered to be as filterable agents because they are able to pass through bacteriologic filter. They are even much smaller than bacteria and we cannot see them using an ordinary light microscope. So the one that you can see here is an example of SARS-CoV-2, the causative agent of COVID-19, and the only way for us to visualize them is to look under the electron microscope. Now, unlike bacteria, viruses could either contain DNA or RNA, but they will never contain both nucleic acid. So the nucleic acid could either be single-stranded or double-stranded. So they are said to be acellular. They are not composed of cells. In fact, many biologists would consider viruses as non-living things. So they are obligate intracellular parasites, which means that they cannot survive without any host cell. So to prevent COVID-19, social distancing is indeed very important because they would require host cell for replication and are usually host specific. So viral emergence become better known nowadays because of the genetics. So we know the DNA and the RNA makeup, the host diseases, signs and symptoms, chemical makeup, geographical distribution. We know that COVID-19 originates from China. They are resistant to lipid solvents and detergents, particularly for non-enveloped viruses. Some of them are resistant to pH and temperature changes, and some of them can be serotype because of the antigenic component. Okay, so let's talk about classification and taxonomy. So when we say taxonomy, taxonomy is the science of orderly groupings organisms into several taxa or taxa, plural, taxon, singular. So taxa are categories. So there are three structured, interrelated categories, so such as classification or taxonomy, and then nomenclature, or particularly the scientific nomenclature or the scientific names, and then of course identification. So taxonomy is based on similarities and differences. So if the similarities and differences are actually based on the genetic makeup, we call it genotype. And in order for us to know the genotype of a certain microorganisms, we have to know the base sequencing of DNA or RNA by means of, let's say, polymerase chain reaction. So here we'll be able to compare the base composition ratio to measure the degree of relatedness between two organisms. Okay? Taxonomy is also based on similarities and differences. Okay, when it comes to phenotype. So what is the difference between genotype and phenotype? Okay, phenotype is the one that you can observe physically, the features of the organisms. And phenotype is actually the outcome, expression of the genotypic characteristics. Now, phenotypes would include the macroscopic and microscopic morphology, the staining characteristics, nutritional requirements, the antigenic markers, physiologic and biochemical characteristics, susceptibility or resistance to certain antibiotics or chemicals. Okay, so because of the taxonomy, we can now actually classify microorganisms. Okay, so we now have three domains. We have the Eukaryote, the Archaic Bacteria, and the Bacteria. Okay, so these are now the three domains. Okay, and because of taxonomy, viruses may also be classified. Now, if you notice here, viruses are not present because, as what I've told you, viruses are considered to be as non-living things. So here, viruses, our viral classification or taxonomy is based on the genomic characteristics, the method of replication, and the virion structure. So there was this International Committee on Taxonomy of Viruses, which can be accessed using this website, and they are the one responsible in classifying viral taxonomy. Okay, now, since this is bacteriology, it is very important for us to know what are the different hierarchy when it comes to bacterial classification. So under the classifications hierarchy, we had kingdom, division, class, order, family, and for bacteria, the family, actually the suffix ends with aci, aciae, okay? And for the genus, it has to be capitalized, and then the species has to be written in lowercase, okay? So species, always with S, either singular or plural. But if we're going to abbreviate it, SP is the singular form, SPP is the plural form. And sometimes there are also some subspecies, and we have to write it whenever it is appropriate. Okay, so here is an example. So for bacteria, there's no kingdom. They used to belong to Monera, but now under the domain, there's no assigned kingdom anymore for bacteria. So phylum, Proteobacteria, class, Gamma Proteobacteria, order, Enterobacterialis, family, Enterobacteriaceae, genus Escherichia, species coli. So we now have Escherichia coli. Okay, so the nomenclature provides naming assignments for each organisms. Okay, in companion textbook. So usually we have the Burgess Burgess Manual for Systematic Bacteriology. So the standard rule, the family name is capitalized and ends in aci, Enterobacteriaceae. Okay, the genus name is capitalized followed by the species or the specific epithet, which begins with a lowercase letter. So the genus species or genus species or the G species, okay, will make up the scientific name. So some genera have the same first letter, so the first syllable is used. So for example, sometimes it's quite difficult to use S kasi Staphylococcus or Streptococcus or Escherichia coli or Entamoeba coli. Okay, so that's the reason why we really have to use the first syllable. Okay, the species would also have subspecies which pertains to the phenotypic differences. So under the subspecies, there could be serovarieties because of serologic differences, biovarieties because of the variation in biochemical test results, and even phage typing because the bacteriophage may actually be utilized in typing the bacteria based on the susceptibility to specific bacterial phages. And then there are also strains. Strains are species with different susceptibility patterns because there could be some strains that are susceptible to penicillin, for example, or resistant to penicillin. Penicillin, that's the proper pronunciation. Okay, then there are also some genetic relatedness because of the rRNA that led to some reclassification. Okay, so 3 domains. Okay, so the first domain, they used to call it Eubacteria because that refers to the true bacteria. Okay, so these are the classical prokaryotic cell encountered in clinical microbiology. This is the one that we will be discussing in this particular course. And then we have the Archaeobacteria. These are bacteria that do not have any cell wall and they are seen in extreme environments. So there are actually 3 types of Archaeobacteria. First is the methanogens that can produce methane gas, and then the extremely thermophilic, which can actually reside in environment that is in extremely high in temperature, such as in hot spring, volcano geysers, okay, at the benthic part of the ocean, and then the extremely thermophilic bacteria. Extremely halophilic bacteria. Okay, those that actually literally love high salt concentration. So this group of bacteria are actually not encountered in clinical microbiology. And then we also have the eukarya. They're actually more complex than prokaryotes and they contain membrane encased organelles. So these are the eukaryotic cells such as the fungi, parasites, and protozoans. Okay, so the prokaryotes are the bacteria and the archaea. And then the eukaryotes are the fungi, protozoans, algae, animals, and plants. So here in medical bacteriology, we will be discussing most of the time pathogenic bacteria, or those organisms that are disease-causing, okay, that may infect eukaryotic hosts such as humans. Okay, so as what I told you, archaic bacteria are those organisms that actually most closely related to eukaryotic cells. So this contains microorganisms that grow under extreme environmental conditions. They are not true bacteria because they lack cell wall, peptidoglycan. So they can stain gram-positive or gram-negative. So cell envelope and enzymes are actually present. It allows organisms to survive in harsh conditions. Okay, so that the ends of the screencast of today's session. See you next time. And this has been Dr. Supachay Basit saying, let's continue to pray because healing is coming. God bless everyone. Bye.